Swayam Prabha. Digital India. Educated India.
The functional anatomy and ultrastructure of the skeletal muscle. The ability to use chemical energy to produce force and movements is present to a limited extent in most of the cells of the body, but in the skeletal muscle fibers it has become dominant. It is through the control of movements the human mind is able to express itself in the form of speech, in the form of writing, locomotion, expressions, etc. So, these skeletal muscles are the muscles which are attached to the bones, to the skeleton of the body. There are other two types of muscle which are called as the cardiac muscle and the smooth muscle. The skeletal muscles are long fibers which are arranged in the form of bundles within itself. In turn, within the bundles there are many numerous muscle fibers which are otherwise called as myocytes. So, all these bundles of fibers and the muscle fibers, everything are arranged parallel to each other, and this is the muscle fiber is the basic structural and functional unit of a muscle. So, the relationship between the individual skeletal muscle and the muscle fibers within itself is something similar to a nerve, and the nerve which is containing many axons within itself. So, similarly here also in skeletal muscle fibers there are bundles within each other, and within the bundles there are many muscle fibers within itself. So, these muscle fibers are actually enclosed within many connective tissue coverings called as epimycium, perimycium and endomycium. So, the whole skeletal muscle is covered by a layer which is called as the epimycium, and this epimycium divides the larger muscle into individual compartments which are called as bundles, otherwise called as fascicles. So, these bundles of muscle fibers are covered by the next connective tissue layer which is called as the perimycium. Within the bundles of or fascicles there are individual muscle fibers, and each individual muscle fiber is connected by a layer called as endomycium. So, basically there are three layers: the outer epimycium, bundles which are covered by perimycium, and the muscle fibers which are covered by endomycium. So, these layers, the epimycium, perimycium and endomycium, join together beyond the muscle belly to form a rope like tendon or broad flat sheet like aponeurosis. So, these tendons are nothing but the connective tissues which are basically made up of collagen and elastin fibers, and these tendons attached to the bones are responsible for the, so whenever the muscles are going to contract, the tendons are responsible for producing the movements by moving the joints by which they are attached. Other than this, there are numerous blood capillaries and blood vessels and nerves which are actually supplying the skeletal muscle fibers which are present within these connective tissue layers, and basically these extensive blood supply is required to produce enough ATP to produce energy which is required for the muscular contractions, as well as the neurons. The skeletal muscle contracts only after the signal which is arising from the neurons are supplying the skeletal muscle fibers through the neuromuscular junctions. So, these neurons are the somatic motor neurons which are arising from the anterior horn cells of the spinal cord, which comes in synapses. So, for each nerve fiber there are individually an extensive arterial supply as well as a venous supply which are present within these connective tissue layers of the muscle bundle. The skeletal muscle fibers are formed from undifferentiated mononucleated cells during the embryonic stage, during the fetal life, which are called as myoblast, and after birth there are presence of another group of cells which are called as satellite cells, which are responsible for regrowth of the muscle fibers due to any trauma after an injury, or also due to the exercise, proliferation of the muscle fibers due to exercise. So, basically there are two types of cells which are myoblast and satellite cells which are responsible for growth of the skeletal muscle fibers. So, these myoblasts are similar to the stem cells, and the process of formation of the muscle fibers from the myoblast are called as myogenesis. So, initially from a single mononucleated cell, that is a stem cell, they proliferate to cause increase in the number of myoblasts. These myoblasts then starts fusing with each other to form multinucleated cells, and they also increase in the in terms of number of nuclei, they also increase in terms of length of the cell. So, this multinucleated increase in length of the cell, the proliferated cell, are called as myotubes, and later there are incorporation of all the cell organelles, the mitochondria, Golgi apparatus, endoplasmic reticulum. Of course, in muscle fibers they are called as sarcoplasmic reticulum with many nuclei. So, comparatively in myoblasts, the nuclei, the precursor cells, the nuclei are more centrally placed, whereas in myofibers the nuclei are more peripherally placed and they are compressed towards the plasma membrane of the muscle cells. So, the individual muscle fibers are larger cells, they are longer in length, each muscle fiber, the length of the muscle fiber will be equal to almost the length of the entire skeletal muscle. They can extend up to 20 centimeters in length, and the diameter of the muscle fiber according to the type of the muscle, it varies from 10 microns to up to 100 microns in diameter, and the nuclei is also very much peripherally placed with numerous mitochondria present within itself. So, next is the group of satellite cells. These satellite cells are nothing but the same myoblasts which do not or which did not differentiate into muscle fibers during the fetal life. So, those remaining myoblasts remains within the surface of the muscle fiber between the sarcolemma, which is the plasma membrane of the muscle, and the basement membrane of the muscle fiber. They are enclosed in the muscle fiber itself. These are called as satellite cells, otherwise called as myosatellite cells. So, these satellite cells normally remain quiescent in nature, that is they are subtle, they do not participate in any other function. During this quiescent stage, the satellite cells has got only few organelles, but have got higher nuclear to cytoplasmic ratio, and they have got enormous amount of substance called as heterochromatin. When the satellite cells become active, the organelles increase in number and the nuclear cytoplasmic ratio reduces, and the number of the amount of heterochromatin also reduces. So, when do these satellite cells become activated? So, these satellite cells become activated following an injury to the muscle. Also, the satellite cells activated when there is a exercise, the muscle is severely acting. So, those conditions also to increase or to cause growth of the muscle cells or to cause repair of the injured muscle cell, the satellite cells become active. So, once they become active, they act in a similar way as to that of a myoblast. How the myoblast differentiate itself from a mononucleated cell and then form a multinucleated muscle fiber, in a similar fashion the process of differentiation occurs and new muscle fibers are generated from the satellite muscle cells. So, these generating new muscle fibers, they undergo the same process of fusion and differentiation to form a myofiber. This new muscle fibers can join with the damaged muscle fibers to cause the growth of the muscle fiber, or they can individually replace the lost muscle tissues. So, the satellite cells can repair the muscle tissue only to a certain extent. If the muscle tissue is severely damaged, then the amount of satellite cells which are present will not be enough to repair the entire amount of the loss of muscle tissue. So, in those cases the degenerated muscle tissue will turn itself into a scar tissue, and of course, there will be a limited extent of function or mobility of the cell. So, the muscle fiber or the myocyte, which is the functional unit of the muscle, is a long cylindrical tube extending up almost the entire length of the muscle, and the classical feature is a striated appearance, which are nothing but the presence of alternating dark and light bands present throughout the length of the skeletal muscle fiber, and also presence of extensive nuclei, hence that is why it is called as a multinucleated cell. So, this nuclei, and along with that there are more amount of cytoplasm as well as mitochondria to meet the energy needs. So, these nuclei are responsible for the protein synthesis. There are many muscle proteins which are involved for the muscular contraction as well as for genetic expression within its local domain, that is wherever it is present. In an average adult human, about 40% of the body weight is contributed by the weight of the skeletal muscles, and out of them 20% of the skeletal muscle weight is actually contributed by the proteins which are present in the skeletal muscles. So, even in an average man weighing about 70 kilograms, about 5 to 6 kilograms is contributed by the amount of muscle proteins present in the muscle. So, that much is the amount of or extent of muscle proteins in the skeletal muscle fiber, and actin and myosin are the most common muscle proteins out, and the third most form of muscle protein is the titin. There are many muscle proteins which are involved in the contraction and regulation of the contraction of the skeletal muscle. The contractile proteins which are myosin and actin, and the regulatory proteins which are troponin and tropomyosin, and the attachment proteins like titin, nebulin, desmin, dystrophin and alpha actinin. So, the contractile proteins myosin and actin are mainly responsible for the generation, or they are the basic contractile machinery of the skeletal muscle fibers. So, myosin is a thicker filament and actin is a thinner filament, and the troponin and tropomyosin, which are already bound to the actin filaments, which are the thin filaments, are responsible for keeping the actin myosin interaction under inhibition, and uncovering of the active sites of the actin is responsible for the actin myosin interaction, thereby leading to contraction. Other proteins which are also involved or indirectly regulating the contraction of the skeletal muscles are proteins like titin. So, these titin are nothing but they are responsible for the elasticity of the muscle. They connect the Z disc to the M line in the sarcomere, and they are also responsible for the passive tension which is generated in the skeletal muscle, that is at any length of the muscle. The amount of force or contraction is actually due to the actin myosin interaction. So, even in the absence of actin myosin interaction, some amount of tension is always recorded from a skeletal muscle fiber, and this passive tension is due to the presence of mainly the titin, which contributes to the elastic properties of the skeletal muscle fiber. And there are other proteins like nebulin. Nebulin is responsible for keeping the thin filaments in a regulated fashion, and desmin is a protein which is going to attach the Z disc, the sarcomeres, to the extracellular matrix to keep the muscle in perfect size and architecture during the contractile machinery. And dystrophin is also a similar protein which is getting attached to the extracellular matrix and the sarcolemma to maintain the structural integrity of the skeletal muscle. And the alpha actinin keeps the thin filaments attached to the Z disc for regulation of its function. So, like this, many proteins are involved in the regulation of the skeletal muscle function. So, in the sarcomere, in the middle of the sarcomere, there are presence of the thick filaments which are myosin. This myosin is then parallelly arranged by the thinner filaments which are called as the actin. The actin are attached to the Z disc on either end of the sarcomere, and the bandwidth which is corresponding to the myosin, the extent of the myosin is called as the A band, and where there is no actin myosin overlap. So, only where the thicker filaments alone is present, that is called as the H band in the sarcomere, and the M line is the one which is bisecting the myosin into two halves, and this M line contains many proteins which are responsible for anchoring the myosin or the thicker filaments together. And in between these are two successive sarcomeres. So, in between that is from the portion where only the thinner filaments are present, beyond the thicker filaments on either side, the portions where only the thin filaments are present, that band is called as the I band. So, this is how the sarcomere is divided into various regions, and when the sarcomere is undergoing contraction, when there is a contraction, the actual size of the actin and myosin or the length of the actin and myosin do not change. It is just going to be overlap of the actin and myosin filaments. The actin filaments, the thin filaments, will move towards the M line. There is change only in the H band and as well as I band, because the actin filaments are moving towards the M line, whereas the A band, even during contraction, the length of the A band remains constant in a sarcomere. Considering the first, the most common abundant muscle protein, which is the myosin. The myosin filaments, there are about 100 myosin protein complexes which are present throughout the thicker filaments. The tail end of the myosin filament pointing towards the M line and the head end pointing towards the Z disc. So, each myosin filament consists of a tail, the hinge region and 2 globular heads. So, within each myosin filament there are totally 6 protein complexes, 2 heavy chains and 4 light chains. These light chains are arranged towards the neck region, and the heavy chains start from the globular heads and then they intertwingle with each other and they extend throughout the tail of the myosin filaments. So, in the globular head there are presence of ATP binding sites as well as presence of actin binding sites. So, the heavy chain region of the globular heads participate in the actin interaction and cross bridge formation, whereas the light chains are mainly having a regulatory function. So, binding of ATP will cause ATP hydrolysis and followed by the cross bridge activation, actin myosin interaction and contraction. So, the thick filaments and thin filaments are always arranged in a regular pattern. Like each thick filament, the myosin filament is surrounded by a regular pattern of 6 thin filaments, and each thin filament is in turn surrounded by 3 thicker filaments. So, this is how it is arranged in the within the myofibril, thereby giving a regulated classical striated appearance when looking under the microscope. So, coming to the other types of proteins, especially the contractile proteins, next is the thin filament, which is nothing but the actin filament. It is called as F-actin or filamentous actin, which are the 2 chains of actin which are intertwined with each other, and these are formed due to the presence of the globular actin molecules which are globular in shape, and the globular heads, the monomers, join together to form the filamentous actin. And there are about 300 to 400 actin molecules present in a single thin filament. And the other proteins like tropomyosin, troponin as well as nebulin. So, nebulin is a protein which is responsible for regulation of the length of the thin filament or the actin filament. Other than that, we have the tropomyosin and troponin molecules. Tropomyosin is again a 2 chain polypeptide which is present in the groove of the actin filaments throughout the length of the actin filaments, and the length of a single tropomyosin molecule corresponds to about 7 globular heads of the actin filaments. And the function of the tropomyosin is that it actually covers the myosin binding sites of the actin, and during the skeletal muscle excitation, the uncovering of the tropomyosin, that is the tropomyosin will move away from the covering site of the actin, thereby facilitating the actin myosin interaction. So, the tropomyosin keeps the actin myosin interaction under inhibition. And there are other protein which is called as a troponin. There are 3 parts of troponin molecules: troponin I, troponin T and troponin C. So, this troponin I again keeps the actin myosin interaction under inhibition, and troponin T is that part of the troponin binds to the tropomyosin molecule. So, when a calcium binds to the troponin C part of this troponin molecule, there is going to be release of this inhibition of tropomyosin by means of signal from the troponin molecule. Thereby the tropomyosin will move away from the inhibition site. The active sites of the or the binding sites of the actin are exposed, thereby causing actin myosin interaction. So, the ratio of actin, troponin and tropomyosin in the thinner filaments is about 7:1:1. For every 7 actin globular molecules, single actin globular heads, there is 1 troponin and 1 tropomyosin present in the entire length of the filamentous actin. So, the troponin and tropomyosin keeps the actin myosin interaction under control, and they are stimulated whenever there is a arrival of nerve signal and excitation of the skeletal muscle fibers. So, next is the presence of the sarcotubular system within the each myofibril of skeletal muscle fiber. So, this sarcotubular system consists of mainly the T tubules and the sarcoplasmic reticulum within the sarcoplasm of single muscle fiber. So, these T tubules are nothing but they are the extensions of the plasma membrane or the sarcolemma of the skeletal muscle fiber. So, these sarcolemma extend deep into the muscle fibers and they run transversely, and their main function of T tubule, they are actually communicating with the ECF. They are nothing but the extension of sarcolemma. So, what they contain is actually ECF, and they are responsible for the passage of the action potentials which are generated in the plasma membrane of the skeletal muscle to penetrate deep into the muscle fibers and causing the excitation mechanism, excitation contraction mechanism. So, this is the basic structure of the sarcoplasmic triad. So, which consists of the sarcolemma, the plasma membrane, and the extension of the sarcolemma deep into the muscle fibers, which is called as the T tubules, and these this sarcotubular system in turn consists of all the sarcomeres which are responsible for the contraction with the sarcoplasm. And another important structure is the presence of sarcoplasmic reticulum, which is nothing but the endoplasmic reticulum of any other cell. So, this sarcoplasmic reticulum consists of longitudinal tubes, the long tubes. At the end there are elongated structures called as cisternae, and there are sarcoplasmic reticulum present on either side of the T tubules. The presence of two sarcoplasmic reticulum along with the T tubules together is called as the sarcoplasmic triad. And the next feature is the presence of the receptors. One is the dihydropyridine receptor present on the deeper invagination of the T tubules, and this dihydropyridine receptors are nothing but voltage sensitive calcium channels which are going to respond to the action potentials which are arriving through the T tubules. And next is the presence of ryanodine receptors. The ryanodine receptors are nothing but the calcium release channels, and the interaction of these dihydropyridine receptor and the ryanodine receptor is responsible for the calcium pulse, which is going to be the enormous amount of calcium which is going to be thrown out of the sarcoplasmic reticulum into the sarcoplasm. So, this calcium which is released is going to cause the interaction, actin myosin interaction, stimulate the cross bridge cycling, and thereby leading to the contraction of the skeletal muscle fibers. So, this calcium, once the skeletal muscle contraction is over, the remaining amount of calcium will be pumped back into the sarcoplasmic reticulum and it will be stored, so that there is no more amount of calcium is available in the cytoplasm, thereby leading to inhibition of the contractions, the actin myosin interaction, or there is a detachment of actin and myosin leading to stoppage of contraction or relaxation of the skeletal muscle, thereby making the process complete. So, this is how the sarcotubular system, which is very much developed in the skeletal muscle fibers, is responsible for the contraction of the skeletal muscle fibers. So, in the skeletal muscles there are presence of other proteins, the dystroglycan complex, which are responsible or which serves as a transmembrane linkage, which are going to link the extracellular matrix to the intracellular cytoskeletal proteins which are present here. Especially the dystrophin, which is responsible for connecting the actin filaments to the syntrophin and desmin, thereby, and in this dystrophin is in turn connected to the dystroglycan through the sarcoglycans, which is in turn connected to the laminin proteins which are present in the extracellular matrix, thereby whatever force which is being generated through the actin myosin interaction is transmitted to the extracellular matrix as well, so that the muscle can function as a unit to cause or to generate the necessary amount of force for producing the particular movement. So, these proteins are contributing individually for the contraction of the skeletal muscles, especially mutations in any of these proteins like dystrophin can cause severe instability because of the loss of connectivity between the extracellular matrix and the intracellular cytoskeleton, can result in severe pathological conditions called as muscular dystrophies. Thank you.